The Curious Case of Benjamin Button was an incredibly successful film that earned over $300 million in profit. It also shed light on the genetic disease Progeria, a premature aging disease, which is the unfortunate reality for some 350 children around the world. However, a new genomic technology called base editing might be able to change that. There are 31,000 known point mutations, which are errors in single DNA nucleotides, which cause genetic diseases, including progeria. That's where gene editors come in. Base editors are able to chemically change one DNA nucleobase into a completely different one without breaking the DNA backbone. Now, you've probably heard of CRISPR-Cas9, and are wondering why it can't be used to fix genetic diseases like progeria. Well, the issue here is this. If you think of CRISPR like molecular scissors, then you can think of base editors like a molecular pencil, which has a much more niche use case of being able to fix point mutations and really small errors in DNA. Because of this niche use case, base editors have a much higher efficiency level than CRISPR-Cas9 does. In fact, base editors have a 40 to 50% efficiency in cell lines compared to CRISPR's only 4 to 5%. Also, they're a lot less error-prone than CRISPR is because they're able to change a single nucleotide without causing a double-stranded break. On the flip side, with CRISPR, after its scissor-like mechanism goes in and causes a change, it's much easier for bases to be inserted or deleted, which can alter the gene sequence and have unintended consequences. The first base editor was able to chemically convert a GC base pair into an AT base pair at a targeted genomic location, and it was developed by Harvard researchers. Later on, another Harvard researcher was able to apply the same principle in order to transform AT base pairs into GC base pairs. This specifically is how a genetic disease like progeria must be corrected. Benefits of base editing include the fact that it's very safe and efficient, although hurdles include the fact that it cannot be used at all genomic locations and for all base pair conversions, as well as, since it is a new technology, practicality on the large scale is still being assessed. ApoBec1 is a naturally occurring enzyme that is able to convert Cs to Us. Us are a type of RNA nucleobase that can be thought of as the counterpart to DNA's T base. Alexis Comer, who is a Harvard researcher, fused this enzyme from rats to an impaired version of the Cas9 nuclease, the one used by CRISPR, in order to make it so that it was unable to create double-stranded breaks, which are a lot riskier. When the guide RNA now directs this ApoBec1 and Cas9 fusion protein to the target location, it is able to convert the C to a U. Then, the cell's DNA repair system takes over in order to convert the impossible UG match to a UA pair, and then ultimately to a TA pair, which is what we wanted. We're not quite there yet, though. This technology needed some additional refinements in order to make it even better than it already was. The Cas9 nuclease was replaced with a Cas9 nickase, which made it so that only the G-containing strand would be cut every time. As a result, the cell would only replace the G instead of the U when correcting the mismatch, leading to a lot more efficiency in the process as a whole. Now, for the other side of the story. The same principle was then applied to create an AT base editor. However, the catch was that there was no naturally occurring enzyme that could accomplish this task, making it substantially harder than it was to create a CG base editor. Through some evolution and protein engineering, the bacterial enzyme TAD-A was evolved so that it could accept the DNA substrate as well as work better in mammalian cells. The resulting base editor was called ABE 7.10, and theoretically, it could fix half of all known point mutations. CBEs, or cytosine base editors, have been shown to correct point mutations associated with Alzheimer's as well as cancer in both mouse and human cell lines, with a 35 to 57% efficiency, compared to CRISPR's measly 0.1 to 0.3%. In addition, the second class of base editors, called ABEs, have corrected point mutations for both sickle cell anemia and progeria, even being used in a mouse model of progeria. In this mouse model, mice that were injected in the second week of life had the best results. including reduced progerian RNA and proteins in several tissues, increased expression of the normal lamin A gene, as well as their histology of their aortas becoming near normal. For mice treated a few weeks after birth, there was an increase of 78% in their lifespan. All of this is absolutely incredible and goes to show that we are closer to curing progeria than we've ever been. Base editing is the technology of the future, and by encouraging its development, we can improve the lives of hundreds of children around the world who suffer from progeria and other point mutation-related genetic diseases by curing their diseases completely, all through the use of a genetic search and replace tool.